majored there in human genetics, so I'm a human geneticist. And there I got a research, I was involved in research programs and got a job at the University of Heidelberg. And I went to medical school beside my research time. Greg said that we did genetic research. I know just about every zoo in Europe because we did research on animals, and especially on apes and monkeys, genetic research, and that was the lineage towards gene therapy. We had in 1976, it was the kindergarten of gene research, gene therapy, stem cells. At that time, nobody talked about stem cells. We knew that we were dealing with some overwhelming cells which had an overwhelming possibility and potency, but we didn't say that it were stem cells. Later on, we did more and more research, went down to the chromosomes, to the genes, and that was the beginning and the beginning of treatment of first of all animals, then later patients. And in my own clinic and office, we are treating patients since 1982. In 1980, I was associate with another colleague, and my own office I started clinic I started in 1982. And it's what I wanted to tell you is a little bit about stem cells, where they come from, what they are, and then in a second part, what we are using it for, and then also part stressing extra medications derived from stem cells, and that should about it. And then after that, I'm open to any kinds of questions. We say in Germany, a sermon in the church shouldn't last longer than 15 to 20 minutes. It's gonna be a little bit longer than that, but as soon as I see somebody dropping his eyes, I will stop. So, stem cells. We are made of semen and eggs, and when they come together, they start out building individuals. And in the beginning, the first 3 months, 12 weeks of gestation, we call the lumps of cells embryos. And the earlier you collect those cells, the more they are able to do. So they are from one cell cloning. We can the sheep dolly. Maybe one of you, most of you heard about it. From one cell, you can make. If you divide the first cells, you can clone whatever how many individuals you want. The longer the gestation goes, the more they are specialized, and we have in the embryonic stage, we have then a lineage which goes to heart cells, brain cells, or gut cells, or whatever, skin cells. And so we have omnipotency in the beginning and a pluripotency later on. That means we can, in the beginning, we have cells which can be everything. And later on, we have cells which can be, which are going in a certain direction, like say skin or connective tissue or heart or brain, neurological, neuronal cells. And we use those pluripotent and or potent cells for a special, from a special tissue to treat different kinds of diseases. The main theme today is cancer. So I will go to that. But yeah, first of all, to understand why we use, as Greg said, at the moment 99% of the cells used are from animals, and we use they are different animal types, but we use, for example, also sheep. But in the beginning, the humans are, when they are in the womb of the mother, they start out as fish, and then they get a frog, and then later on they get humans. So they make in 9 months they make the evolution, and those cells have certain active genes. And in the beginning, for example, about the sheep, we have 99.9% of the active genes in the embryonic stage are the same as the humans. Maybe the genome project, you know, that was a big political issue, and in the United States, billions of dollars were spent for that. And we found already years ago, before that, for example, the chimpanzees, which are our cousins, they have from the gene subsets, they are about a bit 98.5% identical to human, even in the adult stage. We don't take adult cells. Why? Because they know who they belong to the ape, or to a sheep, or to a rabbit, or to a human. In the beginning, they don't know where they belong to. They know where they're gonna be. For a heart, for a kidney, for a liver, for a lung, for brain cells. But that is organ specific. So that's why we can use those cells, because they don't know if they belong to a sheep or to a human or to a rabbit, that they are going to be a brain cell or a liver cell. And if we would collect it later in the gestation, they would know, hey, I'm going to be, I'm a brain cell, but I don't belong to that human. I belong to a sheep or to a rabbit. And then we would have allergic reactions, adverse reactions. And actually the founding father of the new cell therapy was Paul Nihans. And he found that was in the early years of the 20th century, and he published a lot of papers and books about that. And he found that the earlier you take the cells, the less side effects you have, the more effects you have. He started out with a woman who they took out the thyroid, and she got really sick. She almost died. So he took from a fetal sheep the cells from the thyroid and injected that cell to that lady, just to try it, because he said, well, otherwise she will die. That lady recovered, and she lived from that proven more than 50 years after that. So that was actually the beginning that there is something special about those cells. And he did thousands and thousands of people with different diseases, and he found the more you can be very specific in the cell lineage on one thing, but you cannot stress it too long in the gestation. So you have to take it in the embryonic stage or early fetal stage, which is in the beginning, the beginning of the second trimester, a trimenon. There are the cells are absolutely antigenic. They know what they should be, but they have no allergic reaction to that. And we are doing that since 1976, and since this time we never had any adverse or allergic reaction of that. So, and that's imagine how many allergic reactions you have to aspirin. So it's sensational. Why? Because it's biological. The cells know I have to be this tissue, but I don't know if I belong to a rabbit or a sheep or whatever. And we also did a lot of research with pigs, because a lot of heart valves are taken from a pig. But since we have a lot of patients from the Arab countries, they have a problem with pig. So we left the research on pig tissue and took others, that is sheep or rabbit. So, and we talk here about so-called xenotransplantation. That means from a different individual or a different species. And to show how safe it was, about the degenerative diseases, well, the life ends always deadly, normal. So, but we should try to prolong that. So when we start out, when we were born, our genetic subsets is so that we normally could be quite a few years older than 100. But that's also the telomere lengths. With each cycle, they get smaller, and when they are gone, we are degenerated and die. But we try just about everything during our lifespan to cut that short, our lifespan, due to whatever one is doing, or also the environment, of course. So what we are trying to do is, on one thing, to help regenerate the normal body metabolism, but the cancer treatment is something special. Why? Because everybody in the world produces tumor cells every day. It's something normal. Like if Ford produces cars 100,000 a day, there will be a few which they have to throw away. They don't get through the control. So everybody of us is doing that. But in a normal, healthy individual, the immune function is recognizing those cells and is killing it, taking away. And in some cases, in tumor patients, either there are different ways why, but the immune function isn't correct, so tumor cells can develop. And the normal treatment we have so far is cutting down on the tumor cell number of the tumor mass. That means we have surgery, we have chemotherapy, we have radiation. And nowadays we have antibody therapy. The colleagues here know it very well. So we see we have always, except the antibody therapy, we have only therapies which cut down on the immune function, which suppresses the normal regenerative process in the body, which is sometimes necessary. In our practice, we combine sometimes the therapies. But if you just only hit a person or a person's immune function, it's definitely not able to be sure that all the tumor cells are gone, because some tumor cells can hide, some tumor cells can be dormant, sleeper cells, and those are the cells we cannot check because the metabolism is really run down. We cannot attack it by chemotherapy, we cannot attack it by radiation. So those cells slip through. And if I have a reduction of a tumor mass, and I look after that, then I have a low tumor burden. Then, if my immune function is really high, natural killer cells, T cells, B cells, they are more able to recognize those cells when they wake up, and they can catch them. And that's the kick-in for cell therapy, for growth factors. And then there is, because we have there immune restoration, immune modulation, and in some cases immune stimulation. Why? Because in some cases, because we have the stimulation is makes only sense if I have not a disbalance from suppressor cells, which we have too. If the suppressor cells are high in the number or percentage and I push them up, I make just what I don't want, because then I suppress everything else too. So I have those possibilities to modulate, to stimulate, to alterate my immune function. And so the body is able to help part itself. The other thing is I have the possibility with stem cells to do that. But now must be always, there is the question, what happens stem cells? I read in the newspaper or on TV or in the internet, there were stem cells can cause cancer. And in each sentence, there's always a little bit truth. And so we had to look after that. And there are publications where they could prove that from a certain certain amount of stem cells, they were benign, not cancer cells, but tumor cells. They just grow. And if they looked at it, they were all cells which were older than embryonic cells. And in the United States, you have a lot of good scientists, and they do a lot of good things. I'm a member of the American Society of Clinical Oncology. And we have every year a big meeting, about 30,000 doctors from all over the world. And there the new things are published. And the University of Chicago published about 2 years ago a finding that in embryonic cells, you find a protein, they call it Lefty. It's a lab name. And this protein is only found in embryonic stage. In the embryonic stage, which means in the first 3 months of gestation. In the fetal cells, you don't find that anymore. So, and what does that Lefty make? That is recognizing defect cells which have active oncogenes in it, and if you have active oncogenes in that, then they are turning on the apoptosis, which is called the programmed cell death. So those cells go kaput. They are destroyed. And kaput is one of the German words in English. And in the later stage, you don't find that anymore. So in that stage, it would be possible if you were unselected, do a cell therapy, a stem cell therapy, that if you don't look after those problems, you could promote any kind of cell growth. That's why we use embryonic cells for that, and we stimulate with organic growth factors or organ peptides, which are just from the liver, from the kidney or whatever. Because even if you have a liver cancer, the liver is like that big, and even if only this much is healthy, you can stimulate those cells and attack the other ones. So that's why we always in the biotherapy, biological tumor therapy, we always want to that the body helps himself as much as he can. So we treat on one side, or the people are treated with all kinds of chemicals, but on the other side we try to boost the healthy cells with that. And now you can ask, well, how do you know that they don't boost also the tumor cells? That's done. We did a lot of research on that, and it shows, and it was always shown that the subset of so-called repair genes are in those cells, embryonic cells. They are so high that we extract that. I just after that I tell you how we do it. That they are the cells which are already intact, which are healthy, they are pushed a little bit more, that they can, for example in the liver, also detoxify and grow. The doctors here know if you cut out from a mouse half of the liver, after a lifespan of that mouse, they completely regenerate the liver, and the liver grows. That's one of the only organ which and also the kidney, they can grow again. But you have to stimulate, you stimulate those cells by itself. Now, about the tumor cells, we cut down on that. We have, and then we stimulate the immune function, the natural killer cells, and that is something special. The women and the modern fathers who attended a birth, they saw the navel cord. And the navel cord is, excuse me, okay, like that, thank you. The navel cord, in the navel cord there is a part of the connective tissue. It's called the mesenchyme. And in that, in those mesenchymal cells, they are especially active for regeneration, for activation of natural killer cells. That's why the mesenchymal cells, the most research in human stem cells is done with mesenchymal stem cells from that mesenchyme, from that part of the connective tissue. And we use it to, in combination with other extracts, to stimulate the immune function, especially natural killer cells. Also in degenerative diseases, we stimulate a lot with that mesenchyme because it's the starter for repair and regeneration. And now how we produce it. We take cell lines, we shoot at those cell lines with ultrasound, so we crack the walls. And when we crack the walls, we don't need that, that's scrap for us. So we take the ingredients, and with ultracentrifuge we can measure what kind of organelles or growth factors or peptides we want, and that's what we collect. So in a shot we can combine a lot more of those growth factors, of peptides, to boost what I want to. So that's an addition for the cell therapy. We can make it even more specific. And and I want to stress that the mesenchyme growth factors and mesenchymal cells are one of the most important parts for for treating degenerative diseases and also cancer. And we found that doing that, that we even could break chemoresistance. We could break through chemoresistance, which with patient came and showed that they had this second line, third line, fourth line chemotherapy, and the tumor cells mutate pretty fast. So they are you go just to a limit, and that's it. So we tried, and we succeed in most of the cases that we can break through those resistances, and we can go back to chemos if necessary, that they work again. And that was not in our lab, but that colleagues did that they found that even when you do this procedure, you even can cut down on the amount of chemotherapy, so it's not that toxic. That's about the therapy for cancer cells. The degenerative, like for example, we treat a lot of Alzheimer's, we treat a lot of polyneuropathies from different kinds. Everything what's degenerating, we try to first of all we look for causes. A lot of things. It's environment, heavy metal, chronic virus diseases, very important. Lyme's disease. That's and I don't know if it's an issue here. It's getting more issue here. It's in the East Coast. It's a big issue. I know that, and we have it in Germany too. And we come from the southern part of Germany. There, I was grown up. I was brought up with Lyme's disease. That it's for us it was normal. But we found that the Lyme's disease is getting more and more aggressive, because also those, like the tumor cells, the bacteria who promotes the Lyme's disease mutate too. So they get resistant to the normal treatments. So, and that's also chronic degenerative. So with normal chemotherapy or whatever, penicillin or other penicillin types, antibiotics, that's the word, you more and more you get resistant bacteria for that. So with the cell therapy, we can not only cut down, because as I've said before, cut down on the numbers through stimulating or modulating the immune function, but also to cut down on the side effects, which are bother the most patients. We have a lot of patients who said, we had cancer, we were treated by chemo or by radiation or by surgery or combined, and what should I do now? So I said, well, you have 2 or 3 possibilities. One is just to wait until you get a relapse. No, I don't want to do that. So then can't you treat me? I said, well, I don't know. Why not? Because I have to look at you individually. I have to look for how your immune system is, how the function of your organs is, and then I can decide how I can help you. So just rinse everything, take a whole bunch of cells or cell extracts and rinse it over everybody who had cancer. Doesn't make any sense. So it always, in some cases, we say, well, at the moment you are okay, but we would like to check, let's say, in 3 months. We have a lot of prostate cancers, and the sad thing about it is that a lot of, especially surgeons, they say, well, we cut it out and now you're fit again. That's not the way. And the younger generation knows more about tumor biology. They say, well, we cut out the cancer, but we would stress chemo, adjuvant chemo, so that we can be sure. But there is a book, Chemotherapy Heals Cancer and the Earth is Flat. That doesn't make too much sense. So, because when you have a bunch of tumor cells, and the tumor cells are actually dumb cells. They forgot what they should be. They forgot what they had to do. They just can reduplicate, like rabbits. And but not all cells at the same time, and that makes the problem. And so they reduplicate about a third at one time. So I hit that third of the tumor cells with a chemo, but the rest. One is like a boxer, he's not lying on the ground, but he is tumbling. But until the next chemo, they recover. But I still have about a third who didn't get anything from the chemo. So the next time, another third. From the next time, another third. And so you can count. So we cannot get that old that we get rid of all of them. That's mathematically not possible. So we still need help. And that we need that help from biological principles, like regeneration, like immune modulation, immune stimulation. No? Wait. When we do that, we always monitor it, and in most of the cases, we can do some natural stuff, herbal stuff sometimes. In most of the cases, we use cell therapy. Wie viel Prozent war hier mehr? How many? 80, 90. About 90% of the patients we see in our office, we find some kind of defect where we can actually treat and help. A young man, wie alt ist der Herr Böhm? 45 years old, 45-year-old man. He was now 7-8 years ago diagnosed with a glioblastoma, brain tumor, stage 4. He was operated on, but only a small part of it came out. The rest, it stayed. Because it was close to important brain parts, they could not radiate on it as they wanted. So they did a little bit radiation on it. And then he came, and he is still living. And the tumor at that time, from that time on, stayed. We have a so-called stable disease. We see now scars, and the MRI is always the same. And the radiologist always, they write, we see some masses, but from the history, it cannot be an active tumor, it must be scars. And that's now for the last 7 years. And the doctors know about stage 4 glioblastoma. You have a survival time without chemo, without radiation, without operation possibilities anymore. From let's say 4 to 6, at the best 9 months. That's it. And he is living now. And his wife pushed him to retire. He's arguing about that, but as the weaker gender, we obey. So yeah.
Doctor,